Good morning, everyone. Thank you so much for coming today, and I'm really honored to be here and to be a recipient of the Maltz Prize and to be able to tell you a little bit about the research that we're doing at UCLA. So I'm going to be talking a little bit about neuroplasticity and schizophrenia, and specifically how we measure it in the lab and what these measurements might mean. And I'll tell you a little bit about ongoing studies that we have. As you'll hear in Dr. Marder's talk today, managing psychotic symptoms is very important for good clinical outcomes in schizophrenia. However, psychotic symptoms are not all that well related to community functioning in people with schizophrenia. And by community functioning, I'm referring to outcomes such as maintaining employment or going back to school, living independently, and maintaining good social relationships. And as Dr. Green mentioned in his talk, the work in our lab has really been focused on studying determinants of good functional outcome in people with schizophrenia. Dr. Green's work has been critical in sort of increasing the recognition that cognition is a key, important aspect of schizophrenia, and that it's a key determinant of functional outcome. Cognition explains a good amount of the community functioning in people with schizophrenia. Recent estimates place it at about 25%. This work has really served to orient the field towards cognition as a treatment target to help improve community functioning in people with schizophrenia. Dr. Green and Dr. Marder, along with their colleagues at UCLA and elsewhere, and in collaboration with the National Institute of Mental Health, identified the key domains that should be assessed in studies of cognition in schizophrenia. And they developed a compilation of tests that can be used to assess these different domains. The domains that are important include speed of information processing, the ability to pay attention and to sustain that attention over periods of time, the ability to hold on to bits of information in mind and to work with it over brief periods, the ability to learn new visual and verbal information, reasoning and problem-solving abilities, and social cognition, which Dr. Green spoke about in his talk. Our lab has described the pattern of cognitive impairments in people with schizophrenia. Over here, I set the zero line to represent the average performance of a large sample of adults who do not have a diagnosis of schizophrenia. And the blue bars represent average scores for a large sample of young people who are pretty early on in the illness, so within the first couple of years. And the orange bars represent performance of a large sample of adults who have a much longer duration of illness. And these data come from Bob Kern from our lab. And along the bottom are the various domains that I mentioned before. And as you can see, as a group, impairment is evident across domains, although there's a lot of variability within individuals with schizophrenia. And the impairment is also present early in the illness course. My work in this area is moving towards trying to better understand the processes that may underlie cognitive impairments or contribute to them. And one process that we've been focusing on recently in our lab is neuroplasticity. But before I get into why I think it's important for us to study neuroplasticity in schizophrenia, I'd like to provide you with a little bit of background information first. So neuroplasticity refers to changes in the way that neurons respond or the way that they're organized or connected with each other. And these changes may be due to a change in the amount of neurotransmitter that's released at the synapse or the junction between neurons. It could be related to changes in the number of receptors that are available to accept the neurotransmitter. It could involve formation of brand new receptors or brand new junctions or synapses between neurons. And these changes may be pretty brief and transient, as is the case with up or down regulation of neurotransmitters, or they can be much more enduring, such as when we see structural changes to the neuron. Neuroplasticity can occur in response to experience. So some of you may remember a study several years ago that found that the number of years on the job as a taxi driver in London was associated with larger brain regions that are very important for spatial navigation and spatial memory. Neuroplasticity can also occur in response to injury. So for example, if someone were to have a portion of their brain removed from a surgery, or if they were to suffer from a stroke, you may see a recovery of brain functions that were lost over time as the neurons are reorganized. Neuroplasticity is believed to be the basis of learning and memory, and this is reflected in the common saying, neurons that fire together, wire together. And animal research tells us that plasticity is impacted by aging, such that younger brains tend to be more plastic than older brains. Animal research has also told us that providing an opportunity for experiences through an enriched environment can also augment plasticity. There's been a tremendous increase in interest in neuroplasticity research, and there's been development of plasticity-based cognitive training programs in an effort to improve cognitive performance for a wide variety of populations. And we actually use some of these programs in our research at UCLA. And regarding schizophrenia, there's lots of converging evidence that the neurobiological systems that are impacted by schizophrenia are also critical for neuroplasticity. And what this tells us is that neuroplasticity is likely to be impacted by the illness, and it may contribute to the cognitive impairments that we tend to observe in many people with schizophrenia. So one reason why I believe it's important for us to study neuroplasticity in schizophrenia is that it can help us to understand a little bit more about the processes that underlie cognitive impairments. And this may point us towards treatment targets that can eventually lead to improvements in community functioning for people with schizophrenia. I think another reason why it's really important for us to study neuroplasticity in schizophrenia is that many of our existing psychosocial treatments for the illness are to some degree or another based in neuroplasticity and rely pretty heavily on the ability to learn. And Dr. Horan's talk will sort of delve into this a little further when he describes his intervention for social cognition. So now that we have an idea about what neuroplasticity is and why we believe it's important to study in schizophrenia, I think the next logical question is, how do we actually go about assessing it? And to be honest, it's been, the methods are quite invasive. So as a result, they've been limited to animal models. So a typical method for studying neuroplasticity would involve electrode implants into the brain to provide electrical stimulation and then to record neural response. So the neural response is recorded before and after stimulation. And you would look for a change in neural response, what I'll be referring to as a neuroplastic change following stimulation. And so it's pretty clear that we need a non-invasive method for clinical research with humans. Luckily for us, some researchers have recently devised ways of utilizing non-invasive electrophysiological methods to assess neuroplasticity in humans. I'll be talking a little bit about one method that uses EEG and visual evoked potentials. But essentially, these methods work in a way that parallels the invasive methods that I described a few moments ago. So the method that we use in our lab involves visual evoked potentials. Visual evoked potentials are a characteristic electrical response of neurons in the visual cortex in response to a particular stimulus. And they're measured non-invasively from the scalp using EEG. The electrical activity in response to the stimulus is averaged over many trials, and then it's plotted over time. And what we get is a waveform with time 0 when the stimulus was presented, and then going out over time. And what we see is the waveform is comprised of various components, these peaks and valleys here. The peaks and valleys will differ in terms of their amplitude, which is the height of the peaks. The task that we've been using in our research involves presenting a visual stimulus to the participant. So in this case, we're presenting a line grading, and we are recording EEG response to that stimulus. And this is our baseline assessment. The next step is to flash the stimulus at a high frequency for several minutes. And this works to stimulate certain neurons in the visual cortex, sort of similar to the electrical stimulation that I talked about in the animal studies. Next, we record the visual evoked potentials once again following the stimulation. And these assessments can occur a few minutes afterwards, up to even an hour later in some studies. And what we look for is a neuroplastic change, or a change in the visual evoked potentials following stimulation. And typically what we're looking for is a change in the amplitude of the waveform of one of the components. And this is believed to reflect experience-dependent neuroplasticity of the visual cortex. So this is a very interesting method. However, there's only been one published study using this method in people with schizophrenia. Our colleague Daniel Mathelon at UCSF was the senior author on that paper. In that study, they found that in a sample of people with schizophrenia compared to people without mental illness, the people with schizophrenia showed a smaller neuroplastic change following stimulation. This seems to suggest that plasticity of the visual cortex may be reduced in people with schizophrenia. However, further study is clearly needed. Our first research challenge is whether or not we can actually replicate these previously published findings. I conducted a study at the West Los Angeles VA with Michael Green and Jonathan Nguyen, where I assessed visual evoked potentials in 30 people with schizophrenia and 19 people without mental illness. And what I found was that there was a significant neuroplastic change in the people without mental illness, but this was much smaller in the people with schizophrenia, again suggesting that visual plasticity may be reduced in the illness, and this lines up with Dr. Mathelon's findings. One thing that I learned with this study is that detecting these neuroplastic changes can be sort of difficult. The effects are small and can be difficult to detect. And so we're working hard to find the best way to study this really interesting method. My next research challenge is to determine whether or not this EEG task measures what we believe it measures. And whether or not a test measures what we think it measures is a question of test validity, or at least one aspect of test validity. We want to know, does our task assess the desired target, and is it associated with the things that we think it should be associated with, the things that we think are important, and the things that we care about. So in our example here, if we believe that the visual evoked potential paradigm assesses neuroplasticity of the visual cortex, and neuroplasticity is thought to be the neural basis of learning and memory, then it seems reasonable to expect associations between the visual evoked potential paradigm and tests of visual learning. We were actually able to test this. So in the study I just talked about a moment ago, we found that within the schizophrenia group, there was an association between the neuroplastic changes and performance on visual learning, such that individuals with higher scores on the neuroplastic change, or they showed a bigger change in response to stimulation, also tended to show better performance on a test of visual learning. In addition, we found an association with community functioning, again, where individuals with a larger neuroplastic change following stimulation also tended to have better community functioning ratings. For my NARSIDE Young Investigator Award, which is currently underway, I received the award in 2015. I'm delving deeper into this question of validity and trying to learn more about the associations between this visual evoked potential paradigm and different aspects of learning and memory. For this project, I'm working with Dr. Green, Dr. Mathelon, Dr. Nguyen, and Dr. Eric Revis to test associations between the visual evoked potential paradigm and various forms of visual learning. And these tests vary in terms of their complexity, from very simple forms, such as a simple motion adaptation task, to much more complex forms of visual learning and memory, such as being able to discriminate between objects previously seen versus novel objects. And what I'm hoping to learn is, to learn a little bit more about the associations between this task and to build up the knowledge base. I think a really interesting open question is whether or not neuroplasticity changes over the course of illness. As I mentioned from the animal research, we know that neuroplasticity changes with age. And we also have some indications that neuroplasticity may be reduced in schizophrenia, but we don't actually know very much about how these effects may combine together. So for example, the effects could be additive, such that people with schizophrenia may be starting out with a reduced level of neuroplasticity, but they're showing the normal age-related decline. Another possibility is that we're seeing some kind of interaction effect, such that the effects of aging and the effects of the illness are combining in such a way to make the decline in plasticity over time much steeper. I'll be able to investigate this question in a project that's actually starting up pretty soon, where I'll be able to look at neuroplasticity over the course of illness. I'll be conducting this study at UCLA with Dr. Green, Dr. Keith Nectarline, Dr. Mathalon, and Dr. Nguyen. For this study, I'll be comparing the Visual Evoked Potential Index in a group of young people who are very early on in the illness course, so within the first couple of years of illness, with individuals with a much longer duration of illness, maybe 10 years or so. I'll also be able to test visual evoked potentials in a non-psychiatric comparison group that is matched to each of the schizophrenia groups. And these non-psychiatric controls will be matched in terms of age and general demographic variables. And this design will allow me to look at the effects of normal aging on plasticity, the effects of the illness on plasticity, and as well as any kind of interaction between the two. And what I'm hoping to learn from this project is more about the trajectory of plasticity over the course of illness. And I'm hoping that this will inform us a little bit about whether the timing of our plasticity-based interventions might be important during the course of illness. Before I wrap up, I just want to briefly mention a really interesting project that is also ongoing in our lab, looking at neuroplasticity as a potential treatment target. For her NARSAD Young Investigator Award, our colleague Carol Jashan in our lab is looking at whether neuroplasticity can be augmented in schizophrenia using neurostimulation. Data collection for that project is currently underway, so stay tuned for results. So I've covered a lot in 15 minutes. I want to just briefly summarize. I hope that I have convinced you all that cognitive impairment is an important feature of schizophrenia. And one of the reasons why it's so important is that it's associated with important outcomes. And as a result, it's a key target for intervention. We also talked about neuroplasticity's associations with learning and memory, the way that neuroplasticity is typically assessed in animal studies, and the reasons why we believe it may be impacted in schizophrenia. We talked about a non-invasive EEG method for assessing neuroplasticity in humans. And early evidence from this method suggests that visual neuroplasticity may be reduced in schizophrenia. And visual neuroplasticity we're seeing associations with visual learning and with community functioning. I want to thank you so much for your attention. Thank you to the Brain and Behavior Research Foundation for inviting me to speak today. I'd like to thank my mentors, Michael Green, Keith Nectarline, Daniel Mathalon, and Jonathan Nguyen. I'd also like to acknowledge my outstanding colleagues at the Green Lab and the UCLA Aftercare Research Program. I'd also like to acknowledge grant funding from the Brain and Behavior Research Foundation, the National Institute of Mental Health, and the VA Desert Pacific MIREC. Thank you so much.
Great talk. Questions for Dr. McCleery? Yes, here's a question. Yeah. She's coming down. Let's see.
Sorry.
Okay.
I'm just curious, how do you control for past and present different medications that people might have been on while you're doing this study, because the medication and treatments are so varied?
Yeah, that's a really great question. And it's something that comes up in really every research study that we do, the medication confound. And the best really that we can do is we can carefully document we can carefully document medications and try to statistically control for that, but that's really the best we can do. In the study that I'll be conducting with the recent onset sample, part of the treatment protocol for that group is a common medication. And because they're so early on in the illness, there's reduced exposure to different types of medications. So that may be one way of looking at it, but it's a question that comes up over and over again for all of our studies.
Can we get the lights just up a little bit more so we could see? But there's a lady back here. Yeah, great.
In the beginning of your presentation, there was the branding, like MCCB was an assessment tool.
Yeah.
And how is that referred to? Like if I were to ask for my son to be assessed, is that a brand name or is it a general testing procedure?
Yes. So the MCCB is a tool that's generally used in research studies of cognition in schizophrenia. It's available through, PAR assessment tools. It is something that you could, if you're planning to bring your family member in for a neuropsychological evaluation, you can talk to the neuropsychologist about which types of tests they would like to cover. You can mention that you would like certain domains to be assessed and work with them that way. That's a great question.
Yes, ma'am.
Yes, are there any stories about this kind of test on young people that may develop schizophrenia later on? Is this something that just happened, let's say around 13, 14?
Right.
Or when they were 5, you could
Right.
say? Like, I'm assuming that if you see it at 5, there's more range for intervention before the illness starts.
Right. Yeah.
Or is this something that just
It
develops?
Right, and I think that's really a key question. And I think in the past, schizophrenia was typically thought of as sort of a late adolescent or adult onset disorder. But over time, we've come to realize that it really is a neurodevelopmental disorder. One of our colleagues that I'm working on for both the NARSAD and Young Investigator Award and the K23, Dr. Mathalon, he's actually investigating neuroplasticity in young people who have been identified as being at high risk for future development of psychosis. So we're still learning more about this, whether or not neuroplasticity is impacted in young people who have not yet developed the illness but may be at high risk.
I'm the guy who asked the empathy question. Okay, while I'm here, I'm going to share. 40 years ago, I had an extremely flawed psychotic process. I would say I'm about 98% in remission. And I asked that empathy question because I've been in therapy and it's helped. Within that time, I was able to get 2 master's degrees and a couple of licenses. I've been able to work as a psychotherapist. But I actually came really for this session because this is very important to me. One of the things, I want to be validated, one of the things that I did for recovery and continue to do is study academic courses for at least 2 hours a day, things like psychology or sociology, and I've been able to do some teaching. Here's the question. Would study itself, academic study, would that cause neuroplasticity, I'm thinking in the prefrontal cortex, and can actually be therapeutic for someone in recovery with a cognitive problem of schizophrenia?
That's a really wonderful question. And I think that in some ways relates back to the animal work about possibly augmenting neuroplasticity with providing opportunities for experience and learning. So I think it really depends on the individual. As I mentioned before, when I presented the MCCB data for people at various stages of the illness, there's a lot of variability in terms of cognitive performance within people with schizophrenia. So For some people, returning to school and doing that kind of work may provide a very enriching environment that may augment neuroplasticity, but it may differ from person to person.
Great. Well, thank you very much.
Thank you so much.
Thank you.